Princeton. Tell us about Intracellular.
Great. Thanks very much for the invitation to be here. It's a really fascinating meeting people with different perspectives, and I'm hoping to get some feedback on this as well. So I agreed to talk about two networks. Possibly that was one network too many. We'll see how it goes. But let's get started. Here's a brief outline, and the first network I'll talk about is the chemotaxis network in E. coli, which I will introduce and describe as a well-engineered solution to the problem faced by cells of finding food in their environment. And it's kind of cool that it uses some nice physics to get better at doing this job. And then I'm going to transition to the second network, which is the quorum sensing network in a different bacterium, V. harveyi, but it's a relative of E. coli. And this system is used by the cells to basically count how many cells of the same species are in the local environment and to decide how to behave depending on whether the cells are at low density or at high density. And what I'm going to stress here is that compared to the network in chemotaxis network in E. coli, this network really looks complicated with lots and lots of feedbacks. And we're going to try to understand why these two networks might be different. That's the idea of comparing the two. And then, as I promised, I'm going to talk a little bit about the challenges ahead in the general field of understanding intracellular networks. So let me start with a movie. This is the part about E. coli chemotaxis, and like many, many things in this field, it owes a great debt to Howard Berg, who's the dean of chemotaxis studies. He's the one who basically tamed E. coli and made it possible for physicists to study this living organism. And I'll play it again. What you're seeing here is these cells. And each cell, it's about 2 or 3 microns long. And what you're actually seeing are these long protein filaments that are attached to the cell. And they're maybe 10 microns or even 20 microns long. And each one of those is driven by a small rotary motor. So a given E. coli will have 5 of these flagella filaments, each driven by a rotary motor. And as you see, you can sort of see that some of the cells that are swimming look like they've got a little bit of a corkscrew behind them. And that is the bundle of these filaments. And the idea is that these filaments, they're already chiral because the proteins have a hand in this. And so if all of the motors turn in a particular direction, they all turn counterclockwise, then all of these filaments nicely bundle together into something, again, that looks like a corkscrew. And as that turns, that propels the cell forward. So that's how it moves. What you're seeing up there looks like a bit of a tangle, like E. coli is having a bad hair day. This happens when one or more of those rotary motors switches direction. And when it switches direction, it can make the whole bundle fall apart. You get a kind of a wild motion, and E. coli reorients. And then the motors start going the first way again, the bundle forms, and E. coli runs off in a new direction. So you can consider this by the name runs when the cell is swimming along, and then one of the motors changes direction, turns around some random direction, and that's a tumble, and then the cell runs again. And so if that was all there was, this would be some means of E. coli basically doing a random walk around its environment. But E. coli has actually had a lot of time to evolve, and it does something that's actually even better than that. And so what it manages to do is it manages to make a biased random walk. And so if E. coli is swimming in the good direction, meaning that the chemical concentration of things that it likes to eat is going up, it will bias those motors to keep going in the swimming direction. So it'll make a long run. And on the other hand, if it's going in the wrong direction, in the sense that the chemical environment is getting less favorable, that will induce some of these motors to change direction, and it'll tumble. So if you look at the trajectory of E. coli over time, if it's swimming in a gradient of something like an amino acid or a sugar that it likes to eat, you will see that it tends to make long runs when it's going up the gradient, short runs when it's going down the gradient. And this turns out to be an efficient means of working its way up the gradient toward whatever that food source is that it's living on. One last time, in case anyone was not watching the beautiful movie from Howard, in fact, these cells don't have a gradient. They're just looking around at this point. But if there was, you would see this bias. So the thing I want to do is, before I actually start giving you a lot of biological names and showing you arrows and so on, I want to think about this. This is an audience of physicists and engineers and very quantitative people. So let's imagine that we had a job, and that was we were going to design an intracellular network that was going to do this, that was going to perform chemotaxis. So what would we need in order to do this? And so the first thing we're going to need is some kind of receptors, right? If we're going to sense the chemical environment around us, we have to have something that takes the information about the concentration of amino acids, say, and transfers that information inside the cell. And so here I'll give you a receptor, and here's the receptor activity as a function of whatever that receptor is binding to, some attractant. Say this is an amino acid. And for here, it's the world's simplest receptor. I'll have it be active when there's nothing bound to it, just because that's the way it works in chemotaxis. I know it's a little counterintuitive, but we'll just get used to that. And so when there's none of this good chemical in the environment, there's nothing bound to this receptor has activity 1, and then, just according to equilibrium binding with some dissociation constant K, as the concentration of attractant goes up, the probability that the receptor is now bound this molecule goes up, and so the activity goes down, and this is just that curve. And you say, oh, well, that's a start. I could start to build a system out of that. Maybe if the cell is at this concentration and it swims a little bit up a gradient, it'll get to a slightly higher concentration. The activity of the receptor is doing something inside the cell, because the binding part of the receptor is outside the cell membrane. The rest of the receptor is inside, where it can talk to the rest of the machinery inside the cell. And so maybe you could turn that difference in receptor activity into some kind of decision whether you're going to tumble. On the other hand, you might say, well, wait, what if the attractant concentration is a little bit higher out here? There's really not much of a response. You say, well, maybe that means that I just chose a bad value of the dissociation constant for the receptor. Maybe if I chose a slightly weaker binding, I'd spread this response curve out over a larger region. Of course, I'm giving up a little sensitivity at this end. And I'll tell you that while this is not a bad start for this group, E. coli can do better than that. And the problem with both of these solutions is that they don't have any gain. That is, if you have say a 1% change of the attractant concentration, you get at most a 1% change in the receptor activity. On the other hand, E. coli has lots and lots of receptors, and it's swimming up one of these gradients. In fact, even given square root n noise, if it swims into a region where the chemical concentration is 1% higher, it can be absolutely sure it's swimming in the right direction. So instead of just swimming, say, 1% farther, it should just make a binary decision, just to keep going. Things are getting better, let's stay the course. And so the question is, how is E. coli going to do better than this, this 1% bias? And the idea is it's going to do it using cooperativity of the receptors. And the idea is now that instead of these receptors working like lone rangers, each receptor is doing its thing, binding the small molecule or unbinding it and sending some signal to the cell, these receptors are going to work together in teams. I mean, really physically coupled teams. So you have now groups of receptors that are yoked together so that energetically they all prefer to be in the same state. Either in this state where they're inactive and nobody's bound to this, that they're active and nobody's bound to this small molecule, or they collectively all like to bind the small molecule and become inactive. And for example, this curve that I've got here shows the response to attractant concentration now with a team of 10 of these receptors. This is just my drawing, but in fact, measurements show that this cooperativity can be as high as 10 or even 20. So there are teams of 20 receptors working together. In fact, this is a lot like the physics of hemoglobin for anyone who took biochemistry a long time ago. Hemoglobin has 4 subunits, and they all like to either collectively bind oxygen or collectively release it. They like to work in these regimes of the curve. The idea is to pick up the oxygen in your lungs and be sure that you've all picked up oxygen, and then when you get to the tissue, dump it all. The same physics, though, is used here not to make a big transition between these two extreme limits, but actually to get gain in the middle. So E. coli likes to operate right here, where a small change in the attractant concentration turns into a big change in receptor activity. And so, for example, with 10 receptors, you can get a gain of a factor of 10. And it turns out that downstream, where the internal signals interact with those motors that are going to change direction, there is additional cooperativity and ultrasensitivity. So really, E. coli is able to do this trick of turning these 1% changes in chemical environment into binary decisions, go or don't go. So looking at this, it looks good, you know, good idea, but there's an obvious problem. Well, what if the attractant concentration isn't in the sweet spot where you get gain? What if the attractant concentration, say, is higher or lower? You know, is E. coli the sort of specialist that only does chemotaxis in a narrow range? And again, that wouldn't make sense, and evolution has figured out a great solution to that, which is simply to change the input-output relation dynamically. So if E. coli is swimming around at this attractant concentration, it uses this response curve and it gets gain. And then let's say this E. coli does really well, it swims up a gradient and gets over to here. Now initially, that shuts off the receptor activity when it's using this curve, and that's good. If things have just gotten better, that's a good sign, you're probably going in the right direction. Keep going, don't change direction. But ultimately, it would like to, continue to do chemotaxis. Things can always get even better. And what it does is it then adapts this curve to become that one, so that once again, it's in the sweet spot of gain. Now, that's a little bit mind-blowing, but you can think, how smart is this thing? It's only 2 microns long. But it turns out, as in many things that bacteria do, it's not very complicated, it's just clever. And the idea is that there's a feedback system inside the cells that has basically a set point for the activity of these receptors. So imagine that there's this reference curve here, and E. coli likes its receptors to be active, say about 50% of the time. And so in the scenario that I just spelled out, where E. coli swims up the gradient, that receptor activity has gone way down. So it's now close to 0, and as a result, because it's below this curve, that is a signal that means modify those receptors. Do something chemically to actually change the receptors, and that changing of the receptors causes their response to adapt. And so as long as the receptor activity is below 50%, this curve will keep moving out to the right as those receptors become more and more modified until finally the receptor activity is back around 50%. But you can see that obviously in this curve, when you're back around 50%, you're back in the sweet spot of gain. And of course, this is a reversible system. If E. coli gets unlucky and swims in the wrong direction, the opposite thing happens. These curves adapt in this direction, and again, always returning E. coli into this sweet spot of gain. As far as timescales, this is all happening pretty fast. So all of this adaptation, sort of for cells that are really swimming up gradients, is happening on the timescale of a few seconds. But the actual responses, the decisions whether to keep going or to tumble, are happening about on the quarter-second timescale. So there's a good order of magnitude separation between the decisions you're making and how long you're remembering your environment. So if things are getting better than they were 3 seconds ago, keep going. They're getting worse than they've been over the last 3 seconds, change directions. I will briefly point out that everything we've talked about here in terms of the features of this engineered system that we would need to do chemotaxis are represented basically each one by 1 or 2 proteins that have very, very clear jobs. So this is, of course, a schematic of the cell. Here is the cytoplasmic membrane of E. coli, and I'll just walk you through the proteins that are involved here. So first off, here is my sort of schematic of a small chemical that the E. coli is trying to sense. This is, again, an amino acid or a sugar. Of course, there'd be lots of these, but I'm just going to show one. And that will bind and unbind reversibly from these receptors. And I've shown them here clustered because indeed that is one of the themes, that they form these actual clusters of receptors. And MCP is just a technical name for how the receptors get modified. They get modified by methyl groups, so it's methyl-accepting chemotaxis protein. But the idea is, first up, okay, the receptor's job, they're going to bind these small molecules. And what they do is they change conformation when they bind the molecule. And that passes a signal to this protein here. So, in fact, when I talk about a receptor being active or inactive, it really means how is it influencing this protein, A, and everything here is key A for chemotaxis. And this is a particular class of proteins that when it's active, it takes an ATP molecule and it strips a phosphate off that ATP molecule. Here, this little phosphate that's been taken from the ATP, and in this case, it can pass it on to another protein. And the question is, all right, so great, we've got this activity system, and I should say key W is here to help hold these proteins together, so it's got a job that's partly gluing these receptors together. Why do I need another protein that's going to get the phosphate from key A? And the reason is because here's the receptors and there's the motor. Somehow you've got to communicate the signal from the receptors to the motor, so you need some diffusible protein to do that. And that's the job of key Y, picks up the phosphate over here, diffuses around the cell, and it binds to the motors, and it's the binding of phosphorylated key Y that tells the motors basically, things are getting worse, let's change directions, try something else. So what are these other proteins here? Well, key Z has a very simple job. Its job is to strip phosphates off of key Y. If that wasn't there, key Y would hold on to these phosphates for about 7 seconds. And the idea is that's way too long. E. coli is trying to update its view of its environment every quarter of a second. So you've got to turn over this pool of phosphorylated key Y every quarter of a second, and that's the job of key Z. And finally, these last guys, key R and key B, their job is adaptation. So when those receptors are not being active enough, key R is able to get busy adding methyl groups to those receptors to make them more active. And conversely, when those receptors are too active, key B's job is to go and remove those same methyl groups and reduce the activity. So it's the balance of key R and key B that sets that fixed point for the system where it's supposed to have receptors that are active around 50% of the time. And the last little, widget here is a little feedback from key A to key B. This isn't necessary to get this system to work, but it does reflect one of the issues that's present in all these intracellular networks, and that is that the numbers of the proteins are fluctuating in time. They're having to be made, and they're made in a stochastically bursty way. And so what do you do if you've got too much key R? Does that mean that these cells are going to be tumbling all the time? Well, it turns out if the system is too active, it passes a phosphate onto key B, which then would compensate for the higher level of key R. So it's a little extra homeostatic mechanism to deal with the fact that this system has to work in a background where all of the component levels are fluctuating. So let me just say the last thing about this, which is that for those in the audience who like condensed matter, this system really starts to look like condensed matter. And so, for example, at a microscopic scale, if you look at the receptors, they form what are called trimers of dimers. This is a little dimer, 2 proteins coming together, and they come together in groups of 3. But in fact, these trimers of dimers are all organized in a beautiful honeycomb structure. And this is a schematic. I don't have time to go through the electron microscopy, but very much the same kind of approaches that Harold was talking about have revealed this exquisite ordering of these receptors on the membrane where they're bound together by these key W proteins, they talk to the key A proteins, and the whole thing, it's not just a bunch of proteins that are just thrown together. This thing has grown in some way, it is self-organized into this structure that then allows for these groups of receptors to cooperate. And remember, they're cooperating beyond these trimers of dimers. Groups of 20 or more of these receptor dimers are all working together in teams. Okay. So, that's E. coli chemotaxis. And I sort of tried to make the point that, everything that you would think of that this system needs is there, and it's there, and a particular protein is doing it. So now, prepare for something different.
Can I ask one question?
You should, because I need to switch talks.
Good. So, that network is present, is the rotors are sort of distributed uniformly over the cell or all on one side?
So in E. coli, the motors are random, so the 5 or 6 can be anywhere. Many bacteria, that's not the case. They will have, say, one polar flagellum that's doing the whole job. Yes?
And then also the network of receptors, is that uniformly distributed over the surface, or is that just not one? Is there some polarity to that?
So the receptors tend to be inserted generically through the membrane, but they diffuse around and they eventually get trapped at the poles of the cell. So what you'll find is that if you look at a typical E. coli, it'll have a big cluster of receptors at one end and a big cluster at the other. That's not necessary for the signaling because the diffusion of these proteins is so fast you could get by with one protein at one end, the motor at the other. But one advantage of having a cluster at each end is that when the cell divides, each daughter has got a cluster of receptors that's good to go and it can immediately start doing chemotaxis. All right, so switching gears, here is another bacterial intracellular network. It's involved in quorum sensing, and let me just say a few words about that. That is that essentially all bacteria that we know about perform some kind of quorum sensing, that they are making some signals, they're secreting them out into the environment, and then they're detecting those signals. And using that information, they're making decisions about which genes to turn on and off. Because the best strategy at low cell density may be very different from the best strategy at high cell density. And so here's a little schematic. Here's a cell. It's got a chromosome. There's some genes on that help synthesize small molecules. These get secreted. But if there are only a few cells in the vicinity, the concentration of these molecules is low and not much happens. But at high cell density, just by virtue of, say, of having more cells, the concentration of these molecules is higher, and using the same kind of receptors, albeit in this case not clustered, that bind small molecules and send signals inside the cells are getting the signal that there's now an accumulation of these molecules which are called autoinducers, because they can turn the cell on and even make the cells produce more of these molecules. And now the cells kind of know that they're at high density, and they will do different things. And so what do? Well, they do lots of different things in response to these quorum sensing signals. Quorum sensing plays a huge role in the formation of biofilms, and in many cases telling cells when it's time to leave a biofilm. Biofilms are basically most bacteria spend most of their time living in biofilms, not in the laboratory because they're a pain to deal with, where they're in the laboratory they live in shaken flasks. But if you go out into a stream, bacteria are forming biofilms on surfaces, which are cells held together by polymers and proteins and DNA. Cells can make bioluminescence. This is one of the cell I'm going to talk about, Vibrio harvii. It's bioluminescent. But importantly, from the point of view of human health, quorum sensing is very often used to regulate virulence. So cells, for example, will not turn on their virulence at low density, but when they're at high density and there are enough of them to mount an attack against a host, that's when they'll turn on virulence. So they're using those quorum sensing inputs to decide when to make you sick. And of course, that has not escaped the attention of people working in this field, that we need new antibiotics, and maybe one strategy, instead of killing cells, is to interfere with their communication by interfering with quorum sensing. So thinking now about this particular bacterium, Vibrio harvii, we're interested in the network. So what's going on? Well, first thing that makes it a little complicated is that there's not one signal, there are 3. So it's actually making and sensing 3 small molecules. It's got 3 separate receptor systems that sense them, but then kind of curiously, it sends all of that information from those 3 receptors down one single pathway, which seems like a loss of information, and I'm happy to discuss that offline. Those are passed by, again, a very similar system of ATP donating a phosphate to the receptor passing it on to some small molecule, but that's not the end of the chain here. That is passed on to another small molecule, another protein, that is a transcription factor. And transcription factor is a general name for proteins that interact with the DNA, turning genes on and off. But interestingly, even though this protein is a transcription factor, it is not controlling the final targets of quorum sensing, all of the many things that the cell wants to turn on and off. It's actually controlling a family of small RNAs. So these are things that get copied as RNAs from the DNA, but never get translated into protein. They are active as RNAs. And in turn, these family of small RNAs go and regulate messenger RNAs for various downstream targets, which themselves are transcription factors, which themselves regulate downstream genes, including other transcription factors. So you can get a sense for where this is going. But just to see how it works, at low cell density, the system is active and the phosphate is flowing down the system. The small RNAs are getting expressed. But as the cell density builds up, the autoinducer signals are there, phosphate activity stops, and the system is drained of phosphate, and now you stop getting the small RNAs. That stabilizes one messenger RNA, makes the protein LuxR, and it's Vibrio harvia, right? What does it do at high cell density? It makes light. So these cells are now luminescent. Great. Okay, so that looks great, right? We explained it, turns it on, turns it off. But what I haven't shown you is that the system is filled with feedbacks. In addition to having all of these the different signals and the different layers of transcription factors, basically everywhere you can imagine there being a feedback, there is one. And so here, for example, between the small RNAs and their downstream targets, there's a perfect click where everybody regulates everybody else in this loop, and these guys regulate themselves just for good measure. And so the question is, since it kind of made sense, you didn't stand up and shout that it won't work, why do we need all these feedbacks? What are they doing there? And so I will take you briefly through some of the experiments that we did to gain insight of this, and then I'm going to basically speculate. So if you don't understand the experiments, that's okay, because I'm going to speculate independent of some of those results. So here we take this network, and of course it's even kind of complicated just to deal with that in experiments, so we try to simplify it. First thing we're going to do is we need a reporter. So as Harold talked about, there are these wonderful fluorescent proteins that the cell makes itself. So now every time it makes one of these LuxR proteins, it's got a little red floor attached to it. So we can look at how red the cells are to know how much of that protein is being expressed. Then we get rid of one of these systems because 3 seems like too many. Even if you want to study signal integration, 2 is enough. So we'll do 2. We don't want the cells making their own signals. That's too complicated. It's good. We can do the chemistry. We can make the signals ourselves. So we can add the signals by hand in controlled amounts. And then we can look at the cells. And we can look at how red they are. And so here is just a little image doing some microscopy. Here's a bunch of Vibrio harvii on a slide. You look at how red they are. You count the fluorescence. And then for each cell, you count, how red is it compared to its size. And you get a distribution of intensities. And these 4 curves correspond to 4 different levels of autoinducers. Like this curve here with the 2 autoinducers and 1 micromolar, that would be high cell density. That would be what the cells are doing at super high cell density. And then you can see here at the other end with no autoinducers, that's low cell density. But you kind of start to see that, hey, in between, when I have just one of those autoinducers, they're different distributions. So it's clear that you could sort of distinguish more than just high cell density from low cell density. You could start to see different regions of cell density. And I think that's a very important part of understanding this network. And indeed, we can hand the cells, or submerge them in any amount of these signals that we want, so we can plot out sort of the whole distributions for any combination, because I'm losing my pointer, distributions for any combination, and we can also look at the noise, how much cell-to-cell variation, in other words, how wide those distributions are. And then we can do mutants, because this is, there's a bacteria, no big deal, knock out a gene, just a week's work. And so what you see is that you knock out genes, and these input-output relations change. And I have to say, you look at this, and it's not like the sort of simple biology where you knock something out and everything's, gone. It's just quantitatively changed. So we really have to think about these input-output systems and all of these feedbacks from the quantitative point of view. The information is quantitative, and if you don't think about it from that perspective, you don't understand what's going on. And so I don't really want you to stare at those. I just want to give you the sense that, well, what we're going to do is we're not going to see quorum sensing disappear when we kill one of those feedbacks. It's still there, it's just shifted over. The responses have changed somewhat. So if we stare at that a while, we learn a few things. So the first thing we learn is that the feedbacks actually control the ratio of those 2 receptors. Remember, the cell's making up these 2 receptors, we've gotten rid of the 3rd one. But as the cells go from low cell density to high cell density, they actually go from having a predominance of one type of receptor, this orange one, to a predominance of the green receptor. So one of the things this feedback is doing is it's letting the cells pay attention to different signals. So in low cell density, the cells are primarily paying attention to one signal, and it turns out that signal is actually a universal byproduct of bacterial metabolism. So they're kind of paying attention to something that all the cells in their environment might be making. And as that starts to build up, they turn their attention to a species-specific signal. So this already gives you some insight that these guys are actually being able to pay attention to different things. It's like you wake up in the morning, what do you first pay attention to? Maybe you're sensing coffee. Is there coffee? And later on, you're done with the coffee, now you're ready for your laptop. You're looking for your laptop. These guys are like, they're a little simpler, but they got the same thing going, that they're paying attention to what they expect to see coming from their environment. One thing that I thought for sure these feedbacks were doing was going to be reducing noise. Negative feedbacks, it's a great way to reduce noise. I have to say that so far we have found no sign of noise reduction. You get rid of the feedbacks, the system is just as noisy or relatively quiet actually compared to many bacterial systems. And that I have to say is still work in progress. I'm not completely ready to give up on that, and there are some more things we can do. But one thing we have found the system does is it takes the response here, this very sharp response to autoinducers, that's the response in the mutants where you've gotten rid of the feedbacks. The wild type response is much more graded. So as you add these autoinducers, these feedbacks are helping the cell sort of slow down and make its response more gradual, less step-like. And I think the idea here is that there's more than one bit of information that's available to these cells. It's not just about low density versus high density, there's a whole spectrum. And why might that be? Well, the life cycle of these guys is not all that simple, actually. They swim around, and they find a surface. If they like where they are, the nutrients are good, they start forming a biofilm. They make a lot of matrix, and they grow. But once they form this biofilm, there's no point making any more matrix. At a certain point, when the cell density is high enough, they concentrate on making more cells. And finally, this has done no good unless those cells can get free again. So they've got to digest the matrix. So there really is a developmental process that these bacteria are going through, and the only information that they're getting from their environment about where they are in this process is coming from these quorum sensing signals. And so the idea is that the cells will follow some trajectory in the space of these signals to know where they are in that process and what they should be doing. So I promised I would compare the two networks. So, they look very different. One, chemotaxis, very clean design, one quorum sensing, lots of feedbacks. So what's going on? First off, it's not about the components. Many of these components are actually homologs. They're related to each other. The receptors, the internal molecules, they're not different. They look the same, they come from the same ancestors. One huge difference is timescales. Well, chemotaxis, the system's got to update on the 1/4 second timescale. The only way to do that is with protein-protein interactions. There is no time to turn on or off a gene, get RNA made, get protein made on those timescales. Quorum sensing is talking about increases of cell population. That's taking on the doubling time. That's like an hour. So there's plenty of time for gene expression. And I think that's really an important difference, that protein-protein networks seem to be hard to evolve, a lot of feedbacks, a lot of widgets within these networks. But let me tell you, gene expression regulation is cheap. Once you've got a transcription factor, adding one more target to that requires just a few base changes in the DNA upstream of that gene, and boom, that transcription factor is binding and regulating. And finally, I think there's some difference in the functional demands. Chemotaxis is doing one thing. It's following those chemical gradients. End of story. Quorum sensing is actually doing at least two things. One, it's learning cell density, and it's got a wide range of cell densities to do it. But the other thing, and I didn't focus on that, is a lot of those feedbacks seem to be poised for the cell to go from high cell density to low cell density. It is just waiting to suddenly it's in a biofilm, everything's good, and suddenly, boom, it's out in the ocean. And it's got to do a lot of things fast. And it looks to me like this came first, and then this was added as an afterthought. Oh, by the way, it would be really good if we could respond quickly going to low cell density. And instead of redesigning the thing from the beginning, evolution just slapped on a lot of feedbacks to make that happen. So, challenges ahead? Well, the main challenge ahead is that those were pretty simple networks by the standards of real biology, right? This is just stolen from the internet, but these are a couple of eukaryotic networks associated with making vasculature or responding to DNA damage. And like, you know, everything there is a separate protein, right? You know, and they're all interacting with each other. So that's going to be tough. It's also hard to obtain real-time outputs. Like one of the keys to the chemotaxis system was that there was a FRET reporter system that was developed specifically for that, and it broke open the whole field. That's not always that straightforward, or to make controlled perturbations, to come in and twist the network. In chemotaxis, you add a signal. In quorum sensing, you add a signal. Not everything else works like that. And finally, there's a lot of cell-to-cell variability that is going on. And so maybe doing these things in bulk is also giving you a biased picture. So finally, how are we going to meet these challenges? Well, there have been huge developments. So there's huge developments in high-throughput data acquisition. Sequencing is changing everything in biology. I think mass spec is starting to change things in a similar way. As Harold mentioned, there's amazing new imaging tools that are breaking open a lot of problems. And what we haven't mentioned so far is genome engineering. Now we can go and make mutants in eukaryotic systems basically with the same facility that people could make them in bacterial systems. And from the point of view of theory, I'm not so sure, but I am personally fond of some of the top-down approaches that were pioneered by Terry Waugh and his colleagues. Instead of trying to figure out what every widget is doing and how it talks to every other widget, you can figure out what the cell is trying to do, study the basic input-output relations, and that's going to give us a lot of insight into what the widgets should be doing. And from that perspective, it's easier to understand what they are doing. So conclusions, chemotaxis, I think it's a model for the engineering prowess of evolution, and quorum sensing is a model for the engineering limitations of evolution. And there are many challenges ahead, but also many new tools. And let me just put up my slide, thank the people in my group and Bonnie's group, and I put my experimental collaborators in gold because they're worth more than gold. So thank you very much.
I was curious, you mentioned that the E. coli go from synchronous to asynchronous to change directions. So how does that fit into your intracellular mechanism where synchronous beating becomes asynchronous and then synchronous?
Okay,
and kind of related to this is your first movie. You said there was no signal, but it looked like all of them are going in the same direction.
So let me answer those two questions. First off, E. coli, the only sense of synchrony in this system is that the rotary motors all have to be going in one particular direction. So it's not like some eukaryotic cells that have to sort of beat their flagella in the right pattern. These guys just rotate. And if they're all rotating in this counterclockwise direction, they'll spontaneously bundle and swim. And the changes of direction come from one or more of those motors reversing directions. So the motors just go. They go this way or they go that way.
So how does that reverse the direction?
Well, you have a bundle of things that are all helical and they fit together nicely. One of them now starts going the other way and it can make the whole bundle come apart. It's really biophysics. And your second question.
Yes, the first movie that you showed, was there was no signal at all, and all of them seem to be going the same.
That's chance. I mean, I think there may be a bias, but those guys are really just swimming around.
Do you have a good understanding of why you need 10 molecules, 5 molecules, what sets that number? And presumably that's a design parameter too.
We're talking now about the proteins inside the cell, or?
For example, yes.
Yeah. No, that's certainly an important problem. What sets the right number of these proteins? So for example, we actually did some analysis of this for the chemotaxis network, and for example, being able to respond very quickly means that you've got to have a huge rate of adding phosphate in order to balance the fast rate of removing it, and that sets some limits on what the numbers are. So I think you can actually, that's a great question. I think you can start to think about not just what is the pathway made of and what does each protein do to the other, but why do I need 50,000 receptors? And for example, there are versions of the chemotaxis network that evolved from the same origin, but now regulate things that are slower, and they use many fewer proteins, many fewer actual copies, because one of the constraints of the chemotaxis that makes there be so many copies is the idea that it has to be very fast.
So there was a graph in which you showed concentration of autoinducer 1 micromolar, sort of copy number, autoinducer 2, 1 micromolar copy number, and then 1 plus 2 equals, when 1 and 2, the total equals 1 micromolar, you get a bigger copy number. Even though the networks are fed to the same channel, how does, if they go to the same place, why does it make a difference?
Yeah, okay, so you have these two types of receptors, and actually the receptors can send phosphate down the pathway, but they can also remove it. They're bifunctional. So what's happening is, if you have no autoinducers, right, then you have phosphate coming from both sides, and your total level is, 1. But if you shut off one of them, there's no phosphate coming from that side, right? But it's still there, removing phosphate. So you get something that would be sort of a half. And if the other one was there, it'd be a half. Now, it turns out they're not quite symmetric. But you really have to have signals that shut off both to have no phosphate flowing through to get to the extreme limit. So it's really, it is actually additive though. Like, that's something we've quantified. It's really just this additive flux of phosphate from these two different sources.
Hi, I was just curious about gliding motility rather than flagellar motion, because there are a lot of bacteria which also use gliding motility versus flagella. So can you explain can this chemotaxis model also apply to gliding motility versus flagella?
Yeah, there are certainly some cases of cells that glide along a surface that are also using chemotaxis, that are also sensing changes in time. But there are also, there are lots of cellular motility mechanisms. Once you put a surface there are lots of ways of moving along it. Some guys shoot out basically, like fishing lines, grab the surface and pull them in. But yes, there are gliding motility, and for example, in Myxococcus uses chemotaxis for that.
I wanted to ask, do they also use phosphate-based systems or phosphate in general?
Yeah, that is fairly general. That seems to be not just in bacteria, but in eukaryotes as well. That's like sort of the main signaling system at the protein-protein level is that you phosphorylate a target and that changes its activity. It's got a negative charge. It can definitely affect the conformation of a protein. So it's very generic.
Another question here?
Hi. My question is, do you know of any bacteria who do collective chemotaxis? With this I mean different behavior when there is many bacteria compared to what you get in the case of a single cell. So let's say if a single cell goes towards a gradient, many bacteria go, for example, want to go away in the opposite direction, something like that.
Yeah. So I know at least one good example of that, and it's actually the same bacteria I just mentioned. So Myxococcus eats E. coli, will kill and eat E. coli, and it hunts in packs. And they will actually move together in groups, and they seem to be doing collective chemotaxis. They also have a wonderful physics behavior that my colleague at Princeton, Josh Shavit, studies, where they move backwards and forwards, and by slightly changing the rate at which they change direction, they go from being sort of hunting around to a system that actually spontaneously phase separates. So it's a wonderful example of active matter where and this is something that's sort of been worked out in the last few years by a few people, Brady at Caltech and others, where these active organisms that are self-propelled can actually phase separate in just the way that you would see in an equilibrium system. And they phase separate and eventually then form spores, and they do that when they've run out of E. coli to eat.
just out of curiosity, I see Yigal Meyer's name there, and I'm a physicist, and I'd like to know what kind of calculation one can do to model these systems. Do you know that? I mean, what has been Yigal Meyer's contribution to this? Could be interesting.
Well, Yigal Meyer is like my brother. We were postdocs together, and we've been working together ever since. And I feel in many ways he's like a second brain. So everything we do theoretically, we do together. A lot of it is, you know, in the case of the E. coli system, the same kind of physics that you would do in statistical mechanics, looking at sort of coupled Ising-like systems. A receptor is active or inactive. That starts to look like a spin that's, you know, up or down. They're coupled to each other, to, you know, ferromagnetically. So a lot of the things that, you know, I've done with Yigal in that context really would look like you were dealing with spin systems. And with free energies, you know, the works. The system is really physics when you get down to the core of it.